change is detected. The integration center processes information and commands the effector to change the variable even more. And that change in the variable detected, integration center processes it, and commands the effector to change the variable even more. So we don't get something that goes back to normal, we get something that goes somewhere. A destination, if you will, or an endpoint, or some sort of event. So that's sort of maybe the other way to think about it, is that negative feedback acts to maintain variables so that they don't change very much. In some ways that's kind of boring, just keeping the status quo. Positive feedback acts to initiate some event or some final action. Very few good examples of positive feedback in our bodies. Why? Because mostly our homeostatic mechanisms work to try and maintain homeostasis, trying to maintain a variable, but sometimes we need to build up to an event. So childbirth, milk letdown, or during nursing are two really good examples of positive feedback system. So I asked you to go through childbirth, parturition, and try and identify, if you can, the components: sensor, integration center, and effectors. So why don't we go through it? What's going on? Well, one, we've got a cervix, and I can't draw this stuff very well, so just kind of deal with me. Actually, it should be head down. All right, baby. We've got sensors. There are plenty of sensors in the uterus. The ones that really have the strong ability to influence this feedback loop are especially in the cervix. And when a baby's head is pushing against the cervix, it's going to stretch. So these are stretch receptors. They're detecting stretch. What actually initiates this ultimate stretch that causes it is actually still not known. But at least at some point there's whatever signal and enough stretch that we detect the stretch. That information goes to an integration center. It happens to be the hypothalamus. The hypothalamus communicates with the posterior pituitary. I'm just going to put post-pit. And I'm going to include those together as both part of the integration center. And the posterior pituitary secretes a hormone, oxytocin. Oxytocin goes into the bloodstream, floats through the body, and when it arrives at the uterus, it has an effect. It causes contractions of the smooth muscle in the uterus. If you contract the smooth muscle of the uterus and start squeezing on that baby, the baby's head is going to be pushed into the cervix. What's going to happen? The stretch receptors are going to receive more stretch information. So we have more stretch information that goes to the hypothalamus, goes to the pituitary, releases more oxytocin, which causes more stretch, and more and more and more. So we look at the strength of the contractions, the amount of oxytocin that's released is amplified. We get more, and then the next wave we get more, and the next wave we get more, and then more, and then more and more. And eventually, multiple factors, powerful enough contractions, cervix dilated enough that you actually get the baby contraction. And yeah, it's powerful. You guys should know now. I'll tell you, I've got a 1-month-old. So exactly December 14th, I was in the hospital watching right in front of my face. It's pretty shocking stuff. All right. So that's positive feedback. We see how it's not maintaining a variable, that it's amplifying the change in the variable. And this time what we have are forces being pushed on the baby. Higher forces, more forces, until some event. Baby pops out, we're going to stretch. Make sense? All right, good. Why is this not normally good for regulating body variables? Is it going to help stabilize a variable?
No.
Keep it within limits?
No.
It's going to bring it to some endpoint. And so there's just very few examples. I think your textbook, does it use blood clotting as an example?
Yeah, platelets.
That's fine, that is positive feedback. The reason I don't use that as an example is it's very difficult to identify the components. So for us it's easy. Well, stretch receptors, that's the sensor. The brain and the posterior pituitary together act as the integration center, figuring out what to do. And then oxytocin's effect on smooth muscle, that's our effector. When you're talking about a platelet, the integration center is like, it's not even the cell, it's molecules outside the cell. It's a little abstract. So that's why I like childbirth as a better example.
Dr. Word,
Yeah.
so the core variable is the dilation of the...
For this one, if I wanted to define the variable, is what's being affected, it's contractions, which is the pressure, it's the force being applied to the baby. So, you know, the variable here, I would say forces applied to the baby, contraction strength of the uterus, something like that.
Thank you.
Yes. You guys are going to be in charge of letting me know what I put my notes, because I'm going to lose on Good. We feel happy about negative feedback, positive feedback? Good. Core concept we're going to see again and again. This figure here is showing another negative feedback system that helps regulate blood pressure. What I want you to do over the course of at least until maybe by Friday, or if not by next Wednesday, just read over it and outline this very carefully as another good solid example of negative feedback. Identify the sensors, identify the integration center, identify the effector. Make sure you can do that. And this is a good place to do it because it's right in the same chapter. Neural and endocrine regulation. We know that the nervous system, we know that the endocrine system are the two main, let's see if I can. Are the two main systems that are control systems that regulate body variables. Because those are our two main control systems, regulating body variables, it would make sense that they're largely responsible for many homeostatic mechanisms. So together, the nervous system plus the endocrine system, we could say are largely responsible for homeostasis. Not entirely responsible, but largely responsible. And mostly, this is because they have the ability to control effectors. And if you need an effector to modify a body variable, bringing it up or down, you need a control system that can tell it, hey, now's the time to be faster. Now's the time to release that hormone. Now's the time to take an action. These are the things that control our effectors. We've got a name for that. We call it extrinsic regulation. And extrinsic regulation means that a particular body variable is controlled by either the nervous system or the endocrine system. And I'm just writing NS for nervous system, and I'm writing ES for endocrine system because it's just too many letters. And that's the definition. You say something is extrinsically controlled, you mean either the nervous system controls it or the endocrine system controls it. I've got to make sure we know what these things are. What are the organs of the endocrine system? Give me just a couple examples. What's that? Pituitary gland, good. Pancreas. Hypothalamus. Hypothalamus does release hormones.
Thymus.
Thymus, yeah, good. So those are all organs that release hormones. The endocrine system is all about hormone release, and actually by definition, I'll put hormone secretion. By definition, if an organ releases hormones, you could include it in the endocrine system. We'll see when we cover the system in a couple, 3 or 4 weeks. There's actually a lot of organs that secrete hormones that we might not consider endocrine organs, like fat. We don't consider fat an endocrine organ, but it actually does secrete hormones. So you could argue, well, it does, maybe it should belong in the endocrine system, or at least it has endocrine function. Do we know what hormones are? Like, what is a hormone? Yeah, yeah.
Is it a protein?
Some are proteins. Yeah, they're chemicals. Hormones are chemicals. And if you wanted, maybe you could say chemicals, or sometimes you'll see this term signaling molecules. Yeah, they're both good. Yeah, they're signaling molecules. Basically define a hormone as a signaling molecule that can tell another cell or tissue or organ to undertake some action. The key with the endocrine system is where those signaling molecules go. So if this is our organ, it will secrete a hormone. Your textbook is going to tell you that it secretes it into the blood. That's fine. It really still secretes it, but endocrine organs are highly vascular. So there will be blood vessels right nearby where it releases it, and then they go into those blood vessels. once into the blood vessel, then they have access to essentially every tissue in your body. They circulate through your body, and then they can also come out of this capillary as long as it's permeable, and the hormone can squeeze out of it. And now it's at another place in your body, and then they'll do what we call a target cell. And as long as that target cell has some sort of receptor that can bind that signaling molecule, we're going to get an effect. Good, that's the endocrine system. Many body variables are regulated by the endocrine system. We've got a whole unit talking about the endocrine system, and then when we do other organ systems, we'll end up talking about hormones that regulate them, the heart, the kidneys especially. Good, nervous system. I know you guys had a huge unit on the nervous system in anatomy, so you all know what it is. Yeah, I agree. So the nervous system, we could say something like nerves directly innervate target organs. So to innervate just means to form a connection with, or form a synapse with, and they do it directly. So in the nervous system, we'll have a neuron, and it has its axon that will extend out to the target cell. What's it release on the target cell? Remember this from anatomy? What's released from the tip of the axon? Yeah, neurotransmitters. Neurotransmitter is a signaling molecule. And then we'll get some kind of effect. Look at these two systems. What do they have in common? Signaling molecules finding their way somehow to a target cell and then having an effect. It's just that with the endocrine system, they have to go through the blood to get there, and then we call them hormones. With the nervous system, the axon brings it directly to the cell and then releases it, and then we call that signaling molecule a neurotransmitter. Sometimes it's actually the same molecule, it's just what type of cell releases it and does it use the blood. So that's an extrinsic regulation. The nervous system or endocrine system is responsible for controlling the cell or regulating the variable. Most body variables are extrinsically regulated. The nervous system does the job, the endocrine system does the job. Sometimes they both do the job together. There are cases that we'll see in this class that the nervous system or the endocrine system are not responsible for regulating the variable. And if they're not responsible, it's referred to as intrinsic regulation. And what is going to be responsible is the cell itself, or the organ itself. So in intrinsic regulation, by definition, you could say the nervous system and the endocrine system not regulating the variable. You could also say something like, we'll just do organ. The organ regulates its own function. The last question in your homeostasis slide was getting at blood flow. And I have some example of blood flow through the skeletal muscle. The vessels that deliver blood to skeletal muscles are regulated by the nervous system. You can make them dilate, you can make them constrict. They are regulated by the endocrine system. Hormones can make them constrict, and the lactone can make them dilate. But if you exclude those factors, the blood vessels that deliver blood to skeletal muscles can be regulated by the skeletal muscle itself. In other words, a skeletal muscle, independent from whatever the nervous system or endocrine system is doing, can have some control over its own blood flow. In other words, it can release little signaling molecules that float over to capillaries or arterioles and make them dilate. That would increase blood flow to that muscle. If it's doing that, if the skeletal muscle is responsible for increasing its own blood flow, we could say that that would be intrinsic regulation. The skeletal muscle is regulating its own function, regulating blood flow to itself, not the nervous system and not the endocrine system. So yes, it could, but in some examples we'll see that blood flow can be intrinsically regulated. The organ regulates its own function. Let me give you another example. Heart rate. There's some movies that do this. The one that always pops to mind for me is Indiana Jones and the Temple of Doom. You know that time? Reaches into the guy's chest, pulls out his heart, holds it up. And what's the heart doing? It's beating, right? Is it connected to the nervous system? Is it connected to a blood supply that's delivering hormones to it? No. It's beating though, right? And it's beating at a pretty regular rate. So that tells us something. It's beating not because of the nervous system, it's beating not because of the endocrine system, it's beating on its own because it has some intrinsic ability to regulate its own heart rate. And we'll see we do the cardiovascular system. There's anatomy, right? The SA node that's responsible for determining heart rate. It has the ability to intrinsically and spontaneously depolarize and start a contraction. And it does so at a pretty regular interval. That's intrinsic regulation. Does the nervous system influence heart rate? Like the sympathetic nervous system? Yeah. And do hormones influence heart rate? Think of like adrenaline. Yeah. So the nervous system can, the endocrine system can, but the heart also has its own ability to intrinsically regulate its activities. That's what we mean by intrinsic regulation. And oftentimes with intrinsic regulation, that's what you find. There's a combination. The organ can do it itself, but it's highly influenced by the nervous system and or the endocrine system. Stomach pH is another example. Stomach pH is held at about 2 or so, and it will do it even if you cut off all the nerves that innervate the stomach. There are cells in the stomach that detect how much acid is there. If they need more acid, they'll squirt out some more acid. If there's not enough acid, or if there's too much acid, they'll stop releasing acid to keep the pH pretty much balanced. You don't need the nervous system for the stomach to maintain its own pH. It'll do it on its own. That's what we mean by intrinsic. Self-regulation, or it regulates itself. Make sense? All right. Ooh, I want one more. I'm probably beating this to death. I can't remember if in anatomy class you talk about capillaries and precapillary sphincters. Been a while. Do you remember in our vascular system, our cardiovascular system, that we have capillaries which blood flows through, and those are the tiniest little vessels that are bringing nutrients out to areas where cells are going to get them. And there are little bits of smooth muscle that regulate the flow through that little capillary bed. Interestingly, those little bits of smooth muscle are not innervated. There are no nerves that innervate them, which means that the nervous system cannot control those things, and most of the time hormones aren't going to be controlling those things. When these cells up here start running out of oxygen or producing too much waste, so the pH is changing because they're releasing acids, they may also release some other chemicals. It's not clear, but those float over and cause that capillary to dilate, and when it dilates, blood can then flow through that. So you could say that a capillary is engaged in intrinsic regulation. That is, this little chunk of tissue with that capillary can self-regulate blood flow. It doesn't need the nervous system to do it. It doesn't need the endocrine system to do it. It can do it on its own. Those are all the examples I have.
Professor
Make sense? Yeah.
So you said most of our body functions intrinsic or extrinsic?
Extrinsic.
Okay.
Yep. And that's the general rule. So the way to think about it is the nervous system, endocrine system, they're controlling everything. There's a lot of times where the organ has some ability to modify or change its own function. And we'll see good examples of this, especially with the heart. Even though it can do its own thing, it has to listen to the boss. Right? Nervous system, endocrine system, more like the boss, and it will obey the commands. But if they leave it alone, then it's free to do what it wants. Yeah, does that make sense? Yeah. All right, negative feedback control of hormone secretion. This is a concept we're going to see in several units. It's a little odd at first. It might seem a little counterintuitive at first, but because we see it so many times, it's worth introducing it in little pieces. So we're going to introduce it now as a little piece, and then later when we have to process it along with a gigantic pile of other information, it won't be so rough to deal with. So some instructors skip this for now because we cover it again. It's just that I find that for some complicated topics, it's better to eat them like bite and then digest it for 2 or 3 weeks and then take another bite instead of trying to eat the whole thing at once, which can be overwhelming. So we'll see this again. It's referred to as negative feedback inhibition. So we know it's going to be involved in negative feedback. The inhibition tells us something. To inhibit something is to prevent it or reduce it, to suppress it. So it's going to be a way to have negative feedback systems operating and to operate in a way that is sort of self-inhibiting. In other words, we'll see something happen, but that thing that's happening is going to make it regress. And if it regresses too much, then it's going to allow it to go out again, and then it's going to come back. So the definition is not going to make sense. I'm just going to write it down. This is from a book. This doesn't mean a whole lot to me. The secretion of a hormone can be inhibited by the effects it generates. This doesn't really make a whole lot of sense, so maybe a diagram. Let's imagine we have an endocrine gland. We know that endocrine glands secrete hormones. And that hormone goes into the blood, and then it goes to a particular effector, or target cell. And then that has some effect on that effector. It releases a molecule. That molecule goes into the blood and might float back to the original endocrine gland. And I'm going to put a negative sign there, and that means inhibition, or reduce its activity. So here we've got a gland releasing hormone that causes an effector to release a molecule that inhibits the original gland. In other words, this gland is causing this thing to be produced, which prevents it from being produced. If that makes sense.
So it's like, are you saying?
It's self-inhibiting. It's, if you want, first it's like, oh, I want this stuff. But then when this stuff starts to build up, it prevents this from making it. And it's like, okay, I'll stop making it.
So that thing has a limit.
Yeah, it does. So if we inhibit this gland, what's going to happen to the amount of hormone it produces? It's going to go down. And if it goes down too low, then it's going to be gone. Now the effector won't have this hormone causing it to release this molecule. What's going to happen to the amount of this molecule present? It's going to decrease. And when it's gone, that negative feedback inhibition is released. And now the gland is free to do what it wants, which is to start secreting more of the hormone, which causes the effector to release more of that molecule, which then can build up, and then ultimately when it gets a high enough concentration, will inhibit it. So you go through this pattern that's going to be kind of cyclical. The gland releases hormones, that causes this thing to build up until it gets high enough, which suppresses this, then the hormone levels go down, then this goes down, and then, ooh, I can release more hormone. So if you looked at the levels of this hormone over time, you might see them fluctuate.
So is it kind of like the blood glucose?
Let me give you a concrete example. And if we look at the concentration of the molecule over time, it might look like that. If you have something like a molecule in the blood that has a kind of a set point value, it increases and decreases over time. That's characteristic of negative feedback systems. So it's an integral part of many negative feedback systems. And it seems kind of weird to say that this thing is suppressing itself, but it does so sort of cyclically. First it's like amping up, and then it suppresses itself. But when it's really suppressed, then this goes down, and then it can amp up again. So it goes through this cyclic pattern. So let me give you a concrete example of where this happens in the body with actual molecules, instead of just rewriting molecule. And I'm going to simplify this a little bit, because we don't need all the complexity. So we'll take the pituitary gland. If you want, you can put ANT in front of it, the anterior pituitary. And one of the hormones released by the anterior pituitary is LH, luteinizing hormone. This is going to be induced. Luteinizing hormone is the hormone that floats through the blood, goes to the testes, and causes the testes to secrete testosterone. Testosterone then goes into the blood and has various effects. Muscle mass, growing hair, deeper voice, and all those things. But one of the things that testosterone does is it inhibits the anterior pituitary. So we could look at levels of testosterone in the blood over time, and we can see that they go up, and as the levels of testosterone increase, the anterior pituitary's activity is being suppressed, and the levels of LH start to decrease. With low LH, the levels of testosterone decrease. Once the testosterone levels go down, we release the negative feedback inhibition. Now the anterior pituitary ramps up its LH secretion, the testes ramp up their testosterone secretion, and the levels rise up again. So if you look at males and levels of testosterone in the blood, you'll see that it cycles up and down over time because of negative feedback inhibition. There's our molecule, that's what's being inhibited. We'll see this kind of thing happen again and again and again and again in the endocrine system as a way to regulate levels of hormones. But if you think about it, this kind of makes sense. Anterior pituitary wants, if you will, to have a certain amount of testosterone present. How does it know how much testosterone is present, whether it's enough or too much, or does it need to make more? Well, if you suppress it, it's kind of a built-in way to say, well, when it gets too much, shut down production. When there's not enough, ramp up production. So the self-inhibition allows that to happen in kind of a, I don't want to say simple mechanism, but how about straightforward mechanism? We good? Negative feedback inhibition. I always think of the negative feedback, and then the inhibition reminds me, oh yeah, we're inhibiting the activity of our original gland or original hormone. Good. Right. The last section on this chapter is on body fluid compartments. It's sort of abstract. It's the kind of thing that in an anatomy class may not be taught at all. I can't remember if you guys have this in anatomy. I don't think it's there. It's basically a way to take the human body and to look where the fluid is. Say, what kinds of fluid do we have? Where is that fluid located? And can we categorize these types of fluids into different large categories? So we're looking at all the water in the body, all the fluid, and seeing where it is in the body. When we do that, we come up with two main categories. One is intracellular. Compartment. The intracellular compartment is basically the space that's inside all of our body cells. So intra meaning inside, cells. I'm going to draw a cell. So here's a body cell, you have the nucleus, and there's the space. See that? Most of the time you won't hear the word compartment, you'll hear the fluid part of it. So you'll hear intracellular fluid. It basically means the same thing, it's just the juicy part versus the space in the cell. Fluid. And you'll often just see it abbreviated ICF for intracellular fluid. How much of our body water do you think is actually inside of our body cells? Big chunk or a small chunk of it? Big. Who says a large amount? Who says a small amount? Yeah, most of our body water is actually located inside of our cells, at least a larger component of it. It's about 2/3. I see different numbers. This is an easy fraction for you to remember. Is yeah. In other words, at any one moment, you took your body, 2/3 of your body water, roughly 65%, is located inside your body cells. That's the intracellular compartment. Second, why is this important? The intracellular fluid compartment is different than the other fluid compartments. And if you think about it from a cell's perspective, the cell wants very much to regulate what's in this environment. What sorts of things are inside cells? Sorry, that's good. I do hunting, so sometimes I shot guns when I should have had ear protection and I haven't, so things I can't hear well. Yeah, organelles are inside cells. Good. Anything else inside cells? You know there's water, right? That's the blue parts of the water in there. What's that? Well, organelles, yeah, that'll be in there. How about enzymes? Yeah, Yeah, absolutely. Lots of enzymes and other proteins. Proteins. I'm going to put some other things in here that you may not have thought about. Here's the big one that we've got to worry about for us. K+. K+ stands for potassium. Potassium ions, and we know we hear about potassium, we have to have potassium in our diet. Most of the potassium that we have is actually located inside cells. Our cells do a really good job of, when there's potassium outside of them, they pump it inside and then store it inside. This is very different than what's going on outside the cell. Outside the cell, yeah, we're going to have water. We don't have organelles out there. We don't have the same types of enzymes. There will be some enzymes. We don't have the same types of proteins. There will be some types. We have very little potassium, but there is potassium. So we have a difference in the types of fluid. Intracellular, very different from what's outside of it, or the extracellular. This is right, extracellular compartment. But again, even though the extracellular compartment is the space, most of the time we talk about the extracellular fluid, since that's what we're concerned about. Fluid, and then we just use that abbreviation, EC. Extracellular fluid has subcompartments, important subcompartments. There are really two that we care about. I'm going to put two subcompartments, and then I'm going to have another group of just like others. So the first subcompartment is called the interstitium. Or if you want the fluid, it's interstitial fluid. Fluid. The interstitium is the fluid space that directly surrounds cells and is not inside blood vessels. So here we have a cell with this ICF. Outside here is going to be interstitial fluid. You can think of interstitial fluid as basically the bathwater. Cells are floating around in, or at least suspended in, a sea of interstitial fluid. This is their environment. This is where they're going to get everything that they need to live. If they need nutrients, it has to come from the interstitial fluid. If they need oxygen, it has to come from the interstitial fluid. This is where they put their wastes. Cells don't exactly poop and pee, but they produce wastes, and when they do that, they just put it in the interstitial fluid that surrounds them. Just dump it outside the surfaces, or dump it outside of the surfaces. Hormonal signals, neurotransmitters, all that stuff actually gets dumped into the interstitial fluid, and then the cells have access to it. The other subcompartment of the ECF is the blood plasma. Usually you wouldn't even see the word plasma compartment, it's just plasma, and then sometimes you'll see volume, but I guess that would actually have a number inside of it. And the plasma volume is the fluid component that's in our blood vessels. So if I put a blood vessel out here, plasma volume. And it's defined by the limits of your vasculature inside blood vessel. What do these things look like as far as amount of ECF that's in the interstitium versus the amount of ECF that's in the plasma volume? Take a guess. How much of your extracellular fluid, that 1/3 of your body fluid that's left, is actually located in your blood vessels? Lots or a little? Is it a lot of blood or a little blood? We kind of have an image in our head that we have a lot of blood, and we do have a significant amount of blood. But of this last 1/3, about 20% is actually your blood plasma, which leaves about 80% for your interstitial fluid. So of all your extracellular body water, fluid that's not inside cells, about 80% is actually the fluid that bathes your cells. It's where your cells live, and only about 20% of that ECF is inside your blood plasma. All right, what can we say about this fluid? Well, how is it going to be different than what's going on inside the cell? There's going to be water. We're not going to have organelles. We'll have some proteins and some enzymes and things like that. Not a lot of potassium. As far as ions go, I'll talk about one more, but that's the big one. If we look at the sodium that's in our body, the salt, the Na, almost all of it, not quite all of it, but almost all of it is in the ECF. I'll put some there as well. There's some inside cells, but there's very little. So already you can just eyeball this. Potassium inside the cell, sodium outside the cell. You're going to see this as extremely important. anything happens to the differences in concentration of these ions across a cell membrane, you are going to have serious problems. Either your brain will stop functioning, or your heart will stop beating, or your muscles will start contracting, or maybe your heart will seize up, or maybe you'll go into a coma. So this seems kind of weird, and we'll get at why we actually have this situation, but big differences between the ECF and the ICF. Another concept that's useful when we have a picture like this is exchange. Things that are in the plasma volume can freely go into, at least most things, the interstitial volume. Some things are excluded. And then things that are in the interstitium can, at least many things, freely go into the plasma volume. Many things that are in the ECF, not all of them, can go inside cells, and many things that are inside cells, not all of them, can go outside the cells. So this is sort of a concept of exchange. Some things in the blood can go out of the blood into the interstitium, and then they can go into cells. Some things that are in cells can go out of cells, into the interstitium, and then they can go into the blood. And we talk about physiology and how we regulate our bodies so that we can maintain a nice homeostatic environment, a nice place for cells to live. We're talking about how do you regulate this interstitium so that it has the right amount of water, the right amount of salt, the right amount of other ions, the right amount of glucose, the right amount of amino acids, the right amount of hydrogen ions, the right amount of all the things that a cell needs to live in so that it's a nice place to live. We don't want things like toxins to build up, urea to build up, or ions to go out of whack. Regulating our interstitial fluid is what many of our organ systems ultimately do. And since there's this constant exchange, and blood is like the common fluid that's flowing through everything, if our organ systems can regulate what's going on in the plasma, and we have exchange, they're ultimately regulating what goes on in the ICF, or in the interstitial fluid, and that regulates the cell's environment. So when we talk about cardiovascular system, respiratory system, the renal system, and even the nerve system and endocrine system, and what it's doing, a lot of it is going to ultimately revolve around regulating what's going on in the blood. Because if you regulate this, you're going to, by default, regulate this, and that's going to make it a nice place for cells to live. Hopefully your cells can be healthy. If they live, the tissue lives, and the organ lives, and then the organ system functions, and then you can stay alive. So you probably didn't see something like this in anatomy with this fluid exchange, and it's kind of abstract, but it's really useful for thinking about why we have these systems and what they're doing. It's really all about making sure the cell has a nice bath to float around in. And we do that by regulating what's going on in the blood. Questions? Yeah.
So you're saying that the organ regulate just the plasma?
Yeah, and
And
think
the
about,
plasma-
let's just take the kidneys for example. You know where the kidneys are located. Can the kidneys regulate the interstitium of, like the fluid in your big toe? Like they're up here, big toe's down there, right? So they can't directly help regulate the salt balance, for example, but they can regulate the blood. And then when that blood flows back out, it goes down, so you can pick up waste and then bring it back up. So by regulating what's going on in the blood, you have a direct influence on the interstitium of just about every interstitial space that a cell might live in. There are a few places that are kind of special places, but mostly that's the idea. Regulate the blood, keep those values within normal limits, and that will allow every little cell that has a watery environment, wherever it is in your body, to have its watery environment regulated. Not too much waste, enough glucose, enough oxygen, not too much CO2, those sorts of things. Others. The other compartments of the extracellular don't matter. We never really talk about them. I'll write them up here. So you saw in anatomy the lymphatic system. Lymphatic fluid is not located inside cells, so it's part of the extracellular fluid compartment, but it's a very small percent. It doesn't really matter too much. And then there's some other ones like synovial fluid in your joints. Your eyeballs have some juice inside them. The cerebrospinal fluid, CSF, that's another extracellular fluid, but you don't really have to worry about that. For this class, these are the only two that I'm really concerned about testing you on. Interstitium, really important, huge chunk of it, and then the plasma volume, really important, decent sized chunk of it. Questions on blood body fluid compartments. Does it make sense as sort of a concept, the way to think about it? It's a little abstract, but useful. Very useful, since this is really what a lot of homeostasis is all about. Regulating this interstitial fluid by controlling what's in the blood. Chapter 2. Chapter 2 covers basic chemistry. You guys have all had chemistry, so it's just expected that you will understand basic chemistry. How much of your chemistry class is devoted to talking about carbohydrates? Like 0? And proteins? Lipids? Nucleic acids? Right, you guys didn't do this, because that's really the realm of organic chemistry. Has anyone had organic chemistry? Some of you guys have, so you have seen these things before, right? I want you to read through this chapter, chapter 2. I'm not going to test you on it, so don't read it in the sense that I need to memorize all these things so that when I'm tested on it, I can get the answers right. I just want you to read through this section, especially on these molecules, these large organic molecules, carbohydrates, lipids, proteins, and nucleic acid, just with the idea that you have some familiarity about what these molecules are like. And then what I do is, in my lectures, when it becomes important, I integrate the information that I would like to test you on into future lectures. But it really, really helps if you read through these and have some kind of a basic feeling, like, oh yeah, carbohydrates, those are sugars, we can break those down for energy. Something like that. Lipids, oh yeah, those are sort of some greasy molecules, Hydrophobic ends, we can make membranes out of them, or we can store energy as a protein. So just that kind of level. Don't memorize it, just read through it, and that will be good enough. And everything that's really important enough to include in an exam, I'll take out and I'll put into a lecture where it's useful. It'll be a lot easier to remember when we give it to you instead of just talking about all these molecules without any context. So read through those. And now, over the next week, wait for chapter 2. Give it a week. This chapter is dedicated to cells and basic cell functions, but the bulk of it has to do with the structure of cells, what cells are made up of, the types of organelles that are in cells, and what these organelles do. When you read the chapter, you're going to see that there's a lot of information in here. I tend to try and boil it down to just the most important components. So when I go through my lecture notes, you'll see just a few of the things that you'll read about. Focus on the things we talk about. So if it's listing for a particular organelle like 10 different functions, and I talk about 2, that's really what I'm concerned that you have a concrete notion of. In my experience, the logic behind why I do this is that more is not better if you forget more, but you remember few. So sometimes I just make a call. I think that most people remember 2 things, but if I have 3 things, only half the people remember any of them, then I just go with the 2 instead of the 3. So we need to know what cells are. What are cells? Let's keep it basic. Just need a basic definition of what a cell is. Cells are, what's that? Simplest, basic unit of life. Simplest living structure, simplest living thing. Yeah, so when we think about our bodies and why we're alive, why we actually have living tissues and living organs and living organ systems, the only reason that any of that stuff is alive is that the cells that make up that structure are alive. We're only alive because our cells are alive. If our cells die, then we die. And usually it's very specific cells that end up dying. Some of your heart cells, if they die, then the heart no longer functions, and if it doesn't function, then you die. So it can be just a collection of a few physical cells that, if they fail, then the rest of our organ systems are going to fail, and then we're going to die. So you have to have some sort of basic understanding of how cells function, how they're made up, because that relates directly to how the tissue functions, and how the organ functions, and how the organ system functions. So that's why we end up talking about cells. basic components of cells, right? We know that cells have a plasma membrane. We know that cells have cytoplasm. And if you haven't taken biology in a while, there's been some changes, but cytoplasm includes organelles. It's cytosol, the juicy part, plus the organelles. And the nucleus. So let's start with plasma membrane and its structure. And I'm going to abbreviate plasma membrane capital P, capital M from now on. It's just accumulation, right? PM with plasma membrane. The way that I want you to think about the plasma membrane is it's a barrier. Of the cell. So in practical terms, all cells have a plasma membrane. All cells have to have a plasma membrane because all cells have to have a particular space that they enclose. If something happens to the plasma membrane, like you puncture a big hole in it, everything that's inside the cell will go outside the cell, and you'll no longer have a living, functioning cell. I sometimes, and I guess I will this time, use the analogy of your house. Think about your house. It has a floor and then walls and a roof. If you've got no walls on your house, what happens to all your stuff? You come home and your Xbox is going to be gone, your TV is going to be gone, all the stuff in your house is going to be gone from your house because you've got no walls. People are going to come and take it. You also find other things in your house, like maybe the neighbor's dog has come in and is sleeping like, in the floor or something. So it forms the limit or boundary of the cell, and it also prevents many things from entering or leaving. It forms a barrier or a sort of protective shell that's going to exclude a lot of things from going into or out of the cell. Once it made up of So there's two components that make up a plasma membrane that I'm going to talk about. The first is the lipid component. And when you see this word lipid, this is oil is probably better. It's not an oil. So I'm putting it in parentheses. The type of lipids that make our plasma membranes are special. They're phospholipids. But they have some characteristics in common with oil. And the good analogy is, you guys have all done this, you like cook meat or like a nice steak in a pan, and then you get really full, and then you watch TV and you forget about the pan, you come back the next day, and it's like this coagulated grease. And then you stick it underneath the water, and you like run water on it, and the water just beads and like rolls off. And if you want to actually clean the pan, you have to like add soap on it. You have to actually add something that breaks it up. So when you think about the lipid component, I actually want you to have in your mind an image of like grease or oil, something greasy. Does grease interact with water very well? No. So think of that as kind of your barrier. Water and grease don't really mix. If you have like a balloon and you rubbed it with like Vaseline or grease and you poured water on it, it would just bead right off. And that's a kind of a good image to have for these types of lipids. The lipids are special. They're phospholipids. Most books will depict them as a circle with a couple of little tails on it. There you go. The tails indicate fatty acids, which I realize probably doesn't mean a lot to you. We'll talk about fatty acids from time to time in this course. Fatty acids are long chains of carbon and hydrogen. They are the main thing that's in fat. So fat is going to be made up of special conglomeration of fatty acids. They are hydrophobic molecules. They are the greasy, oily part of an oil or a grease molecule. They are hydrophobic molecules. What does hydrophobic mean? Yeah, water-fearing. That's not the perfect word. It's what it means. A phobia is to be afraid of something or to have an aversion to something. It's not that they're afraid of water. It's just that water and the fatty acids, water and those hydrophobic portions of the molecule don't interact very well. No, they don't really repel. It's just they don't interact. It's kind of like if you had like a bunch of, like a scoop, and you scooped out butter and put it in water. The butter is not pushing the water away. It's just that the water and the butter don't mix very well. So if you want, you could say it repels. That's fine. But it's better to sort of think, well, it would rather hang out with other fatty molecules. And that's where we pick up next time.